So in this video, we're going to move into a more advanced area of molecular modeling. In the introductory videos, we covered moving the models around, selecting things, doing mutations at specific residue sites, and building a peptide. In this advanced series, we're going to be looking at how do we modify proteins at a broader scale, and in this video in particular, we're going to look at how do we connect the linker or the peptide that we made in the prior exercises to another protein, such as this GFP. So in this series, because we're doing more advanced activities, you do need to have a 3-button mouse. And this is simply because you'll be doing more activities, and a 2-button mouse or a trackpad likely isn't enough. And so that's highly recommended. Okay, so in this video, again, we're going to be connecting the peptide that we created to a terminal end of the GFP protein. Okay, and so similar to before, let's simplify our model. So I'm going to remove waters. And I'm also going to move to 3-button viewing, molecules. I'm going to remove one of these GFPs, and now we can see we have the GFP here, and then we have our peptide, which is currently in its ribbon format. So one of the first questions we'll ask is, which end of the protein do we want to add the protein peptide to? I'm going to go to residues here so I can select certain areas here. So this is the N terminus, the beginning of the protein. And this is the C terminus, the end of the protein. And so perhaps we could just put it up to the end of the protein, the C terminal end here. So what I'll do is I will deselect the N terminus, and I'm going to show the main chain as sticks for that. And then similarly for the peptide, I'm also going to show sticks for the main chain there. All right. Okay. So we can see the end here. Remember, the end of a protein has a carboxylate group. And so we can see the double bonded oxygen here. And then on our peptide, we've got also have a C-terminal end, but then we also have an N-terminal end, which is a little bit hidden. There it is, right up here. And so using a 3-button mouse, we're going to go into editing mode here. And if you want to move a specific object, you hold Shift and use the middle button. In this case, it's a roller button, so I'm going to hold Shift, and I'm going to click on the object, and I can simply drag it. So I'm going to drag that N-terminal end here, the amine end, into close proximity of the C-terminal end here. And I'm going to rotate it a little bit. And then I'm going to move it again a little bit closer, rotate it again so that it's just into close proximity. And that's actually pretty good, because remember, there's actually going to be a bond created between these two. So you don't want them touching. You want to give enough space for an extra bond. All right, so we're still in 3D editing mode, and one of the next things we need to do is we need to select the atoms that we want to connect together. And you can do this while you're in editing mode by simply clicking on the atoms. So in this case, we'll click on the nitrogen at the N terminus of the peptide, and we'll click on the carbon at the C terminus of the protein. And because these are currently two different objects, right? So we have two different files that were loaded. We loaded the GFP and we loaded the peptide. Pymol won't let you connect these two using the simple create bond feature in the build. You can only create bonds within a single object, right? And these are currently two different objects. And so we need to use a different feature which is not listed anywhere, and it's called the fuse feature. And so we're going to fuse, and then we have to tell the program what we want to fuse together. So you'll do a space, and you'll see that the name of what you selected here is PK1 and PK2 in brackets. So we're going to go PK1, we're going to fuse that, comma, space, PK2, bracket, comma, space. And there are actually different modes in which you can fuse them together, and you can find these online, but I'll just tell you, you probably want to use mode number 1, which is kind of going to sync up the amino acids with the other one here, okay, the residues. And we're simply going to press enter, and when we press enter, you're going to see that the program is going to fuse them together, but it'll also create a copy so that it's all together in one object, okay? So we can see here, we still have, so I pressed enter, and we can still see the original peptide here, but now we have, it's created a fused version all within the GFP file, right? And now it's actually asking me if I want to change this bond angle. And this bond angle, we can simply look at it and we'll see here that there really isn't too much steric hindrance here. There's no atoms that are noticeably close to one another. And so sculpting should take care of this, no problem. So I'm just going to click off here. And now we can see that we have our fused peptide and protein. One feature which I didn't talk about earlier is hiding and showing objects. So if it is a light gray up here, then that means it's visible. If you click on it, you can actually hide an object quite easily. And so I did that, and now we can just see the fused GFP peptide linker. All right. Now, one thing you'll notice is if we look at this top bar here, there is, we have our amino acid sequence for our protein here. Sometimes when you're doing this, it doesn't add your amino acids in the right place. It'll kind of just insert them at somewhat random places. When you use mode 1, it's usually pretty good. But let's go through this and just have a look to see. I see here, so when we get to the end, this last one here, it's as though it fused the amino acids with the previous last amino acid, threonine 230. So we don't actually see the residues within this sequence. And so these programs, they often have little glitches like this, and often one of the solutions is to export and import into different programs. So before we get there, let's go ahead and do some sculpting, and then we'll open this up in a different program, and hopefully we can fix this problem. All right, so I'm going to go to viewing here. I'm going to open up the wizard like before. I'm going to click on sculpting, and I'm just going to choose the this atom here right at the edge of our linker, and it's going to start sculpting. You can see there's not too many changes at all. The backbone angles must have been pretty good. Actually, one thing we'll notice here is right at the joint where we linked the proteins together, which is right here, you can see that this nitrogen has 4 bonds. Okay, so it's got 2 hydrogens coming off and then 2 bonds to the neighboring carbons. And this actually isn't really all that possible. You wouldn't normally see this. And so it looks as though when we created the bond, it, for whatever reason, retained the 2 hydrogens on that amine. And so let's stop the sculpting feature here, and we're going to go to all, and we're going to remove the hydrogens under the action. So I was going to remove all hydrogens, and then let's go all, and let's add those hydrogens back. Okay, and so now you can see that it's actually correct. There's only one hydrogen here, which makes up 3 bonds to the nitrogen, so that's correct. And so now that we have the correct structure here, let's go back into sculpting. Let's go ahead and click on the bond there again, and let's let it work a little bit more here. You can see that it is twisting down. All right. Let's just let this go. Yeah, the whole peptide is twisting. There we go. Oh, there we go. This makes sense. These oxygens are often pointing in the opposite direction. So that was a pretty major change. It'll keep twisting here. You can let it run for overnight if you want, and come back and see how it looks the following morning, or you can wait a little while, 10-20 minutes. You might get a close approximation. All right, so I've let it go for about a minute, and I'm just going to stop it here. There was quite a lot of change happening in the linker region here. And so now what we can do is let's go ahead and just have a look at this new molecule here. We can do some of the things we looked at earlier. So for example, we can, let's go show the main chain that sticks. Let's show the side chains as lines here. Let's hide the cartoon. There we go. So that's what this modified protein could look like. So when you're doing sculpting, we talked a little bit about solvation, where you can actually put solvent around your molecule and then do more energy minimization, and I will demonstrate that in another video. That's one thing to keep in mind. Why don't we go ahead and look at the surface. So if we create a surface on this, let's show the surface. There we go. So this is now what the surface looks like, and you can see this is where the tag is. That's pretty cool. If given enough time for energy minimization, or if you're using a different program, this linker might actually curl up onto the surface of the protein. So that's something that can happen. So let's hide that surface. We can also do, there's some other plugins that we can look at here. So for example, the electrostatics, or the charge at the surface of the protein. And so if we just go ahead and look at this polymer, and let's just go ahead and run this. It's gonna, I guess there's a few issues, but let's continue. It'll take a moment to compute. And so this is going to look at electrostatics, or the charge at the surface of this protein. And it'll look at the different amino acid backbones and side chains that are visible or available to the surface, and they might be positively charged or negatively charged, or hydrophobic, so no charge. And so here we go. So we have, for whatever reason, it did not, I guess I didn't select this piece as well here. It's too bad. So let's go ahead and delete this group. We'll go back to here. All right, and so now what I'm going to do is I'm going to export this as a Pdb file, or export it. I think it exports as a CIF file, which is very similar. So I'm going to enable. So these are the things that you're seeing. So that's good. That's what we want. And I'm just going to go ahead and save this in my crystal structures file here. And I'm going to call this GFP C terminal peptide. All right, so now I've got that saved. And in the following video, I'm going to open up that file in a different program, and we'll see if PyMol was able to create a more generic file that can be opened up in different programs. All right, in the last video, we talked about how we can fuse two different proteins together using the fuse feature. Sometimes that can lead to some issues with the program knowing where to put the amino acids in the sequence. And so PyMol is really good for actually adding on peptides and amino acids within the program itself. And as you'll see, it's really quite straightforward. So let's go ahead and do that. So as normal, let's simplify our model. Let's remove waters. We're going to remove one of these chains so that we have a single chain here. And similar to before, we can find the C and N terminus on this side of the protein. So before we had created a little peptide and we had added it to the C terminus. Let's go ahead and add the same peptide using the build feature. Okay. So in this case, you're going to actually let's add or show the main chain as sticks so that we can see where the different atoms are. Let's find the C terminus. So it looks like there's a nitrogen here. So that's the start of the protein, the N terminus. And we have a carboxyl group here, or double bonded oxygen, which represents the C terminus of the protein. And so now what we need to do is we need to identify the atom that we want to build our peptide onto. Okay, and the way we do that is we need to go to 3-button editing mode, and we simply need to click on the atom. There we go. So I've clicked on the atom, and there's a little sphere there. You'll see a little listing. It designates this as PK1. That's the name of the selection. And now it's as simple as using build at the top, residue, and choosing the amino acid. So before we had gone glycine, so I've just added a glycine. Let's add another glycine. All right, now let's add another residue, lysine. And let's add another residue, serine. And the last residue here is tryptophan. All right, so that's that. We can now click on this to remove any selections. We can get out of the editing mode. Here, if we scroll to the end of the sequence, we can see our sequence is right there. Let's go to residue so we don't select the entire chain. And if we select our new peptide we just added on, let's color that something else, maybe pink in color here. We can actually zoom in here and make sure that everything is added properly. So we can see that we had the former C terminus here, and you can see there's a single bond to a nitrogen which has a single hydrogen, and then it carries on and so forth. So that actually looks like it added it nicely. There's no mistakes, and then also within the sequence there's no mistakes here. And the main reason for trying to make sure the program understands the order of the amino acids is that if you export it to another program, which we will later on, sometimes the bonds can be all messed up because the other program doesn't know exactly where to put them. Okay. So as the C terminus, why don't we do the same thing to the N terminus? So PyMol is smart enough to know how to add the amino acids on in either direction. And so we'll do the same thing. We will go to editing mode here. we're going to click on the N-terminal nitrogen. So again, it'll say PK1 here. And we can add on a build residue histidine, all right? Now, histidines or polyhistidine tags are very common for purifying proteins. And so let's go ahead and add a couple of those. All right, so there's one. You can use the shortcuts here if you want. Let's add 2, 3, 4, 5, 6. And you might recall that at the beginning of proteins, you always need to have a methionine there. And so if you were to create this protein in real life, you would have to have a methionine ahead of your polyhistidine tag, right? Because the methionine is the start codon. So I'm going to go ahead and now add a methionine here. There we go. So this, and let's look again here. Let's go back to the beginning. Let's highlight our 6 polyhistidine tag and change the color to, let's say, yellow backbone here. Actually, I probably still have, no, it's all good. And let's get rid of the 3-button editing mode here. And there we go. So now we have a polyhistidine tag at the beginning of the protein at the N-terminus, and we had to put a methionine there because that is encoded by the start codon. And at the end of the protein, we have our custom peptide that we had made before. And this time we didn't have to use the fuse feature, and the program very clearly knows exactly where these amino acids need to go. We can now do the sculpting feature, and you can sculpt either end, or we can broaden the shell and the cushion to include both. But if we reduce these to, let's say, the radius, we'll go 10 and 10, and we click. Let's see what these histidines do first. So you can see it's not quite fully encompassing the entire N-terminal peptide. But let's go ahead and, so that's pretty cool. They're all twisting a little bit. Let's expand this. There we go. So now we can see our other, similar to before, the tryptophan and the lysine residue like to separate apart a little bit. Still moving a little bit. You can see some of the histidine residues are still moving. Again, you can let this model run overnight if you want to on your computer. I'm going to stop it here. I'm going to go to the build and sculpting, and let's see what sort of sculpting options we have here. So there's auto sculpting. You can change the number of cycles per update, so that basically means that it might do 10, run the algorithm 10 times before you see it update. And that could, if you make it so that it'll do a lot more cycles per update, that might, your computer might be able to handle a little bit better. And then there are also some other options here for optimizing bonds or all terms, for example. So we click on all terms, which may have already been the setting. Let's go sculpting again and let it run further. There's no major changes happening, so that, oh, there we go, and now we're running. You can see that there are some motions happening here, but nothing major. All right. So I'm just going to stop it there, and now one thing you can do is with your new protein, you can again save this to a file by going File, Export Molecule, and you're going to want to export the stuff that's enabled, right? So again, on the right-hand side, this molecule, the green fluorescent protein that you've engineered, is enabled, so it's going to only use that. And you may as well just leave these other options as they are, and you can click Save and save to a folder. And then that can be opened again in this program or in other programs. All right, so now I've opened up our engineered protein where we have built on the N-terminal polyhistidine tag here and the C-terminal peptide chain that we had made before. And this is a program called Chimera, which is developed by the University of California, San Francisco. And it's also a really awesome program. It's more powerful than the current open source version of Pymol, but Pymol is very user-friendly and you can do a lot of things in it. And if you export your proteins properly, you can then leverage some of the features of Chimera. And that's what we're going to do now. So I'm just going to go to some preset viewings here so we can see our protein. So you can see our polyhistidine tag here, and then our other tag, and then we have the rest of the protein over here. All right. So one of the features that I really want to use with Chimera is if you go to the Tools, Structural Editing, we can add hydrogens just so that we have them. Click OK. All right, now we have all of our hydrogens. Let's go back again to structural editing. So the main thing I want to use here is the Solvate feature, which will simulate putting some different solvents around the protein. So I'm going to click on that. Bring it back on here. And there's many different solvation methods. So you can put your molecule in a box of solvent, so it'll be like a big cube. You can just have a capping layer where it's a very thin layer. You can choose the radius here. And shell is usually one of the better ones to use. And so when you put in here, that is the depth of the shell. I believe in angstroms. So if we put 5 angstroms here, and then these are the different solvents. So tip 3 is, this one's water. If we look higher, there's some other solvents, right? So you can see like methanol, for example, here. And so let's stay with tip 3 box. And this box here will remove any solvent or ions present and then resolvate it in water in this case. And so now we're going to add, let's apply that, and it's going to calculate. All right, I'm going to close that. And so all of these little triangles are actually water molecules. If we go back to the presets and go atoms here, you can see there's the 2 H's are the whites, and then the oxygen is red. So that's pretty cool. And yeah, so this is more or less what it could look like within a cell, where you don't just have your protein suspended in air or in vacuum. It's actually completely coated in solvent molecules like water. Okay, so if we go to select here, let's select the protein. Let's show the ribbon here so that we can see the protein better. And that definitely helps us to see a little bit more. Let's see if we can select the structure, the solvent. Let's show ball and stick. So now you can see the different balls and sticks are the bonds. But let's show these as spheres. All right, and so you have to remember the van der Waals spheres are kind of the outer edge of the atoms. And so if you were to look at it in that way, you can see that there's actually a pretty packed shell of water molecules now surrounding the protein. All right. And so because we could continue to work in Chimera, but because we're used to working in PyMol, we really just wanted to kind of do this one thing. We've now solvated the protein. We can now save this file as Pdb file, and you can already see I have tried this before. So I've created a file called solvated NNC GFP. All right. And you can do the same thing. You can call it whatever you like. You save that, and then we're going to go and open that in PyMol. All right, so this was what our protein previously looked like. So we have the main body of the protein and then our two tags here. I'm now going to open our solvated protein. And so this is what it looks like. Let me just increase the depth here. So similar, with the difference, again, being the shell here. So one thing I always like to do when setting up a simulation is to remove and add hydrogens. So let's go remove the hydrogens, and then let's go ahead and add back hydrogens. You can see now in PyMol it's not showing the little triangles, it's actually showing water as not having a strange bond between the hydrogens or little triangles. So that's cool. I'm going to go ahead and remove our old molecule here that we had developed earlier. So I'm just going to go ahead and delete that object. So we're only working with this one here. You'll see in the sequence bar there's some craziness happening where there's just O's, which represent waters, in between all the amino acids. And then at the end, if you have lots of O's, that represents all the extra solvent molecules there. But what we can do is we can select our tags here. I'm going to color those. Let's try this. I actually try a different color. Let's color orange, something different. And then similarly at the N terminus, let's go ahead and color those something different. We actually have to deselect these ones or else they'll also change color. And let's do these ones, blue is a little different. There we go. So now we have those. Let's go ahead and add the backbone to all of this as sticks. I can see that a little bit better. And then lastly, let's on our peptides, let's add the side chains so we can see. Oops. See what's happening. Sticks here. All right, so now we can see our peptides nicely. They're surrounded by water molecules. And I'm going to go into the build sculpting. And I've actually changed the setting to update the screen every 1,000 cycles. Things happen a little bit faster on my computer, and then you can also see the changes happening a little bit better. So we'll leave it there. And let's move this to the center. Now let's go to Wizard, Sculpting. We've got a pretty big area here. I'm going to focus on the polyhistidine tag at the N terminus, but it should catch the C-terminal tag as well. So here we go. So you're going to see some movement happening. And again, this is going to be quite slow because it's a laptop computer. If you have a very powerful computer, things will move a lot faster. Just want to twist it here so we can see the histidine residues right in this area here. So we'll just watch this for a moment. It's difficult to see, but the waters are slowly moving around, the side chains and the backbones are moving around a little bit. So I've constrained this a little bit more here. You can see the histidine residue here is continuing to twist away from this alpha helix here. And this will continue to happen. You could probably run this for more than a day and it'll continue to move around because it's dynamic. As you move one molecule around a little bit, that's going to influence another molecule like a solvent. That might move a little bit, which will then influence another side chain, which might make that move a little bit, which will move another solvent, etc. Because you have hundreds of solvent molecules and thousands of atoms within the protein, this will continue to happen, right? And so, as we were talking about earlier, with energy states within cells, like if your protein is folding and it relaxes to its most thermodynamically stable form, meaning it's the lowest energy state, it's relaxed. So this is sort of what's happening right now. These molecules are trying to move around so that they are not experiencing, let's say, repulsion by other atoms, and they're going to continue to try and find a state which is has the lowest energy state. So I'm going to click done here, and again, so this could go on and on and on, and let's turn on the. Let's show the ribbon, let's show the cartoon in the, oh, I already am, in the side here. Let me hide the side chains, because one thing that is happening is this is getting more and more wiggly. It's like the backbone here is it's slowly kind of coiling even a little bit. Okay. So I'll leave it at that for now, and in later videos, we'll explore solvating and editing the proteins in different ways. And you'll see this is a pretty good tool to understand more about the molecules you're working with. And you can use different solvents as well. And if you are to tag two proteins or domains together, you can run simulations to see how they might interact with one another. So yeah, really this is just the beginning of using these tools to explore proteins and their structures.